Okay, I think I'm on here now. I had to like refresh this screen a couple times to get connected. Um, all right, so it looks like we got a good crowd today. All right, so I think we're doing neurophysiology. I had to check the syllabus to make sure we kind of touched on neurophysiology. I mean, we did membrane potentials and action potentials and what happens when they sum and what happens when they innervate a muscle, and that kind of brought us into muscle physiology. So I guess we're going to kind of zoom out, and instead of looking at the molecular stuff like electrolytes, sodium and potassium, we're probably going to zoom out and do a little bit of vocabulary. And I had to pull up some notes just to kind of go over what I think is what we want to talk about with neurophysiology. So before we get started, do I got any questions? I guess we had one of these Sunday, last Sunday, and I thought that was spring break, and that was my mistake. I didn't know we were having these. So if any of you guys were the participants on Sunday and you were left hanging for a while, I think they had to get somebody in there to cover it. And so I apologize on that. But I think I got the schedule back to normal. So let me get the chat going here. We are starting the last week of our lab. Let's touch on that real quick. We're starting the last week of our lab, and that is pretty much the appendicular skeletal system, the axial skeletal system, and then we did bones and tissues and connective tissues tonight. So if you got any questions on that, please bring them up. I know one of my students came up with an acronym for the epidermal layers. So, but maybe everybody else has already had something. But anyways, I think we're okay on that. I don't see anything coming through the chat. So let me try to pull up I want to try to kind of use the notes here. And I'm going to use the notes basically to go over what we really want to talk about with neurophysiology. And the big difference is that we have the CNS and we have the PNS, right? Central nervous system and then the peripheral nervous system. And the central nervous system basically is made up of the brain and the spinal cord, whereas the peripheral nervous system is kind of like the cranial nerves that extend out from the brain, the spinal nerves that come out of the spinal cord, and then of course the ganglia, which ganglia basically means is like these little nodules of cells that will kind of fire an action potential together to make sure we get the signal to where it needs to be. But let's back up and talk about the cranial nerves. The cranial nerves, what really cranial nerves do is that, excuse me, my nose keeps itching. It's like allergies out. What cranial nerves really do is that they help deal with giving us some kind of innervation or feeling in our face, right? There's a trigeminal nerve, which kind of helps us feel our jawline. There is a few other ones that are involved that kind of help feel the face. I cannot think of them right now, but they kind of branch out from the upper portion of the spinal cord, and will I think like eyelids, maybe eye movement is one of them I can think of. Anyways, okay. And then the peripheral nerves, the spinal nerves, basically this is what makes up like the central, if you remember the autonomic nervous system, which is part of the peripheral nervous system, is that it is home to the sympathetic nervous system as well as the parasympathetic nervous system. The sympathetic and the parasympathetic are kind of part of the autonomic nervous system. So what happens is that when these nerve endings kind of leave the central nervous system and become the peripheral nervous system, they kind of go out into the body, right? They can innervate anywhere from our legs to our intercostal muscles while we breathe, our diaphragm, when we breathe. The big ones are like is what happens when we have the sympathetic nervous system. Okay, the sympathetic nervous system, that is pretty much, it will control the smooth muscle of our gastric GI tract. Okay, I guess that's what I'm trying to say. Our enteric system, where if we were to digest food and we have this bolus of food moving through our body, it will help contract and move the food through the small intestinal tract, and then as it kind of goes through, it's when it's called moving, it's called peristalsis. And what happens is it kind of molds it over to where it can take out X amount of nutrients and whatever it needs, and moves the rest of it down into the large intestine, and then that's kind of how we form our stool. So that's kind of interesting. This is kind of the nerves that we don't have any control over them. This is the autonomic nervous system, so we don't have any control over it, and it just moves into the example I just used is basically where it's controlling the gastric, the GI tract. Okay, I'm trying to gastric gastrointestinal tract. That's what I wanted. It's the I keep thinking it was an E. Okay, and then ganglia. Ganglia I want to talk about real quick, because if you remember that we have sympathetic and parasympathetic nervous systems, right? We also have presynaptic neurons, and we have postsynaptic neurons. And depending on which PNS or that's parasympathetic or sympathetic nervous system we're talking about, it really depends where the ganglia are, right? So we have like a presynaptic neurons, and then we have a ganglia, and then we have postsynaptic neurons. So depending on where, if we're talking about parasympathetic versus sympathetic, really will help us determine where the ganglia are. Now, because it's kind of interesting, the ganglia, as I mentioned, are these nodules of cells. They're like a little pocket of cells, right? And they're kind of like the midway point, so to speak, from presynaptic to postsynaptic division of the nervous system. So the parasympathetic nervous system will have a much longer, a much shorter presynaptic neurons. Okay, so the ganglia are going to be a lot closer to the spinal cord, and then the postsynaptic neurons that are kind of after the ganglia are going to be much longer. All right, you can probably see this in a textbook at some point. Whereas the sympathetic nervous system, they have a much longer presynaptic neurons, okay? Much longer, so there's a lot of neuron that travels away from the spinal cord, and then we reach that ganglia nodule, and there's a much shorter postsynaptic neuron. And that's basically just to make sure when we do fire on the sympathetic nervous system, that the target is specifically ready, or the postsynaptic neuron is specifically targets a certain area that we want to fire on. Okay, and that's kind of a big deal, just because that's kind of when we get into fight or flight, right? That's either where we're gonna stand up and make our stand and fight something that we think is threatening, or we run, and we run away from it. So it's kind of an important thing. Okay, so I think that's all I want to touch on with the anatomy part. I want to see if there's anything else through these notes that I want to touch on. Here's something else. Sensory versus motor neurons. So sensory is otherwise known as afferent. Okay, so you may see this afferent term on a test that kind of helps determine what we're talking about with afferent. If we're talking about afferent neurons, we're really talking about sensory neurons. So what this means is that if you were to step on a tack, and this pain receptor will fire back to the brain to helps you acknowledge that yes, you stepped on something sharp and it's painful. It travels through the afferent neurons. Okay, and not only does it travel through afferent neurons, it will synapse into the brain, which stimuli the pain receptors are, and then we will lift our foot up. Well, we can't lift our foot up off the sharp tack if we don't have motor neurons, and these motor neurons are called efferent neurons. E. So sensory is afferent, and then motor is efferent. And these are just vocabulary terms that you're going to have to want to get used to because they will likely be used on an exam. And this kind of helps us figure out what's going on and how we can avoid hurting ourselves. The best example to use is basically a pain receptor. So we have sensory afferent and then motor efferent. I probably should jump on here and make sure I don't have any in the chat. Okay. So any questions there before I move on? I'm going to try to kind of hit the high points of neurophysiology. Okay. Yeah. Thank you, Brooklyn. At least I'm glad you answered. It's better than me just sitting there waiting. Okay. All right. So I have this table that we kind of already touched on with the GI tract and the autonomy of it. Another example of the autonomic nervous system is basically the heart rate, right? And we have talked about heart rate back when we talked about muscles and how we have the sympathetic nervous system and the sympathetic nervous system creating a much faster heart rate, and then we have the parasympathetic nervous system that creates a very slow heart rate. It slows the heart rate down, and that is also part of the autonomic nervous system that will innervate the SA node. And the SA node is also like a nodule of cells that help control your heart rate. It's called the SA node. You'll learn more about it in Bio 139. Okay, let's touch down to nerve cells, right? So we've talked about neurons and how we have afferent and efferent, right? Efferent is sensory, and then afferent If you look at it, sensory starts with an S, A starts with afferent, and then M is motor, and then E is efferent. So sensory has afferent neurons, motor has efferent neurons. So it would spell out like that: sensory, afferent, and then motor, efferent. I think that's the best way, because these vocabulary terms are going to show up on exams, and it's kind of important that that you don't get confused about what an afferent thing is. Yeah, no problem. My microphone cuts out. I don't know why. I think it's a Blackboard Collaborate issue, because I'm literally, as you can probably see through your screen, like I'm on top of this camera here. So I don't know why it keeps breaking up, but I'm glad you all chatted me something. Okay, Caitlin, it may be something on your end. I think my microphone is working. Can everybody hear me? You may have to turn up your volume. Okay. Okay, thank you. Caitlin may have to Turn not sure why. Okay. I am recording it, and that goes for it, guys. I am recording this for YouTube if you prefer. Yeah, so if you want me to throw this up on YouTube, I'm happy to do so. If you want to watch this again, I think a lot of my students recommend that I have some kind of recording outlet to where they can watch it again. So if you're interested in that, just let me know. It could be any of you guys, and then I will happily upload this later on tonight, and you can watch it again. So if you think it's helpful, I may just do it anyways. Who knows? You always get like 1 or 2 views from somebody. Okay, so motor efferent. Okay, cool. Caitlin, if you, I told everybody when you were gone, I can upload this on YouTube, and you can check it from there if you want to hear it again and whatever you think you missed. Just let me know. All right. So I want to go over neurons and glial cells. So neurons we kind of talked about, right? These are the neurons make up the afferent neurons and efferent neurons are all the same. Motor neurons do have a much longer axon, which is very interesting. Motor neurons have a much longer axon, and that just kind of helps propagate the signal farther, closer to the target. You know, like your sciatic nerve, stuff like that. It's a much larger nerve that has to have a lot larger cross section, much greater width. Instead of like this, it'd be bigger. That way it can propagate a signal farther down. And that's kind of like the difference between sensory neurons and motor neurons, is that motor neurons have a much longer axon. But let's talk about the more supporting cells, right? These are like the glial cells and all their roles. And we kind of touched on this before, but I think we should revisit it. So glial cells are supporting cells. They don't have an action potential. They're non-excitable, okay? And they have all kinds of different roles. Like astrocytes, for example, that's something I did my thesis on. And the role of astrocytes with ALS. And astrocytes are kind of like the nutritional cell that will help maintain homeostasis and give neurons the nutrients and support that they need to carry out their roles. And sometimes, more particularly, motor neurons. And when these astrocytes begin to die off, sometimes the motor neurons begin to die off, and then that's kind of how we see this motor neuron collapse, and then we kind of get different neurological diseases form up. ALS is one of them. Not as much Parkinson's. There's other ones that I'm trying to think of that have issues with motor neurons. PLS, which is a lot different than ALS. ALS is more you can die from it. PLS, you live a lot longer with it. And I cannot think of what the P stands for, but anyways, it doesn't matter. Microglia. They used to think that the brain was not was kind of like immune privileged. And what that means is that there is no immune response in the brain itself. And I think over time that narrative has changed. And it's upon the discovery of microglia. And what microglia do is that they are kind of like the cell that picks up all the debris from the brain. Now, there's not a lot of turnover of neurons in the brain. It's not like skin cells or gastric cells, gastric epithelial cells that kind of like die all the time and then come back. There's not a lot of room in the brain to have a lot of cells die off and then come back. So there's not a lot of turnover of neurons. Neurons usually have a much longer and sustained life compared to its other constituents. However, when something does happen, microglia are there to clean up the debris. If there's some kind of traumatic event or something similar to that. Yeah, Gage. That's kind of what I was talking about. There's always been something where, yeah, if you go sniff a bunch of markers or something like that, I mean, you knock out all your neurons, and then you sniff yourself stupid or something like that in school. Some of that's true. I mean, the cells do come back, but like when you're talking about cells that die off, neurons, the microglia are there to clean up the debris, right? If something were to disintegrate, then these microglia kind of help process this debris and kind of find its way out of the brain cavity. The brain does not have a lot of room for for just wasteful stuff like that. Now, Schwann cells and oligodendrocytes, they do the same thing. All right, they do the same thing. And I'm going to try to move this over a little bit so I can see the chat and do this. All right, there we go. So Schwann cells, and like I said, Schwann cells and oligodendrocytes produce myelin. Okay, they produce myelin. And myelin, if you remember, is the insulating material that wraps around the axons. And what happens is that when you have this myelin that you have an axon that's been myelated, it will lower the resistance of the action potential to travel. So you don't have a lot of stuff working against the action potential, right? If we are to insulate something, then we keep the heat in there, or we things are not allowed to dissipate, and we have a stronger signal that gets sent down the line, right? Versus an unmyelinated axon. I think pain fibers are also unmyelinated, and that's why they're slow. I brought that topic up tonight in class, and I said they are slow fibers. The reason why they're slow fibers is because C fibers, otherwise known as pain pathways, are unmyelinated. So they travel a lot slower. And that's why if you put your hand on a hot skillet, it takes a minute to register that your hand is burning, because the pain, the signal that it triggers, the travel to your brain to tell you that skillet is hot, is slow. And that's why that unfortunately with burn situations it takes like literally a split second. It's not exactly like at a snap of a finger for you to determine that yes, something's on fire or something's burning. And that's just because pain fibers, pain efferent, right? Or excuse me, afferent, pain afferent sensory neurons are slow because they're traveling to the brain to tell your brain, it says, hold on, this skillet's hot, your hand's burning, you need to let go. Same with the TAC example we used earlier. So the Schwann and oligodendrocytes help avoid this. Schwann cells are in the peripheral nervous system. Here, that's the difference. The Schwann cells help myelinate the axons on the axons in the peripheral nervous system, while the oligodendrocytes are only found in the central nervous system. Okay, and that's the big difference. They both do the same thing, but you want to be able to determine what Schwann cells live and where oligodendrocytes live. It's an easy question to get mixed up. It's usually, they probably pitch them between each other on a multiple choice list where you'll have Schwann cells as B and oligodendrocytes as C. Most people will pick B, but the true answer is probably oligodendrocytes. So just be aware that both of them do produce myelin, and that Schwann cells are found in the PNS and oligodendrocytes are found in the CNS. Ependymal cells, they are cells that make up the blood-brain barrier, okay? They're kind of part of it. What they really do, ependymal cells do, is that they create cerebral spinal fluid, CSF as we call it. Cerebral spinal fluid basically is a type of fluid that is kind of low in sugar, low in protein. It's kind of like more of a lubricant. water-based lubricant, and ependymal cells will produce it. There's probably about 150 to 140 milliliters of this fluid that will pretty much be contained within the brain cavity, and it's kind of used as a absorber, more like a shock absorber for any kind of traumatic event to the brain. Sometimes we've had cerebral spinal fluid leaks. If you were to, if the cerebral spinal fluid were to become infected, this is kind of how we get. I do this all the time. I'm blanking. I'm thinking of what I mean is I do this test at work all the time to determine if this person has. It's not. Yes, thanks, Cage. This meningitis. I kept wanting to say pneumonia. I was like, that's not pneumonia. It's meningitis. Yes, meninges, meningitis, inflammation of the meninges, which is part of the spinal cord. Thank you, Cage, for that. You saved me embarrassment. So when somehow, I don't know how this happens, somehow bacteria can track up into the spinal cord and brain and can infect the meninges. It's more rare, but it does happen. Sometimes people are more leukopenic, which means that they have a low white count or they're on some kind of chemotherapy agent, and something crazy takes over like a yeast, and you can get it's called cryptococcus. You can get that in your spinal fluid. It's rare, but it does happen. And normally what you'll do a spinal tap, and it comes to me, and then we will do a cell count on it to determine if this is a meningitis event or not, and if it is, whether it's one viral or two bacterial. Bacterial is the more likely to, more lethal one, I guess, as I should say. Excuse me. Viral meningitis, they'll put you on some kind of stuff, anti-inflammatory, and it'll probably subside on itself. Herpes can do this. A lot of herpes simplex can get into the meninges and flare up. Most of the time it's herpes that does this. There's some other ones, and then there's some more serious ones like bacterial. And you've probably seen group B strep is one. Listeria is the big one. You'll see Listeria is a cold growing bacteria that is, in the past, it has made its way into cantaloupe. And when people eat cantaloupe, it can cause Listeria. The big problem with Listeria is that, yes, I've seen it affect people and through meningitis, but it also is one of the rare bacteria that can attack the fetus. So more pregnant women who contract Listeria, either through eating something cold in your fridge or cantaloupe, something like that, it can, start to harm the fetus. So I don't know where. Yeah, I'm trying to remember where we're going. Yeah, ependymal cells. Let's get back on track here. Ependymal cells produce CSF. Now, when I first introduced the blood-brain barrier is a series of cells that block the brain out from the rest of the body. So CSF will be contained within the blood-brain barrier, and when we get above the BBB, as I call it, because if you say it, blood-brain barrier, if you say that 5 times, you won't be able to say it. The CSF is above that, and everything below is something without CSF. If there is CSF, then you probably have something kind of going on. I remember at work, these ladies were freaking out because they had a runny nose, and they thought they had a CSF leak. I said, no, it's just you have a runny nose. It's not CSF. If you did, you'd have a severe headache, and you'd probably, have other things going on. Okay. All right. So let's see. I'm going through these notes right now. I'm trying to talk about something worth interesting. Do I have any questions, by the way? Any questions at this point? Okay. Thank you, Kelsey. Okay, so we're getting all this. All right. There are. I want to go over one more concept, and we'll probably call it a day. I wanted to pull up. I don't think I can share my screen. Yeah, I can. I haven't done this in a while. I want this. All right, you all should be able to see that. I want to go over this picture real quick. I want to orient you guys. This is the spinal cord. This is looking down the spinal column, okay? And there's something I want to talk about. And I'm going to get back on the chat. So we have a posterior view. This is going to be your back. And then over here is going to be your stomach, right? Anterior, that's going to be the front of you. This is going to be the back of you. Now, these are the spinal nerves that I kind of talked about coming off of the spinal cord. This is the spinal cord. The interesting thing about the spinal cord is that you have your white matter right here on the outside, and then you have your dark matter in the middle. All right. Now that's opposite for the brain. The brain's different. The brain has white matter in the middle and then dark matter on the outside. But when you look, sensory afferent neurons will leave the spinal cord on the back end of the spinal cord, the dorsal side. Okay. So if something were to come in, it's going to leave and come out the dorsal side of the column. Motor neurons, all right, motor efferent neurons come to the ventral side of the spinal cord. Okay, it's kind of interesting because it's when you get into anatomy, you start poking around and seeing what things really are. You have sensory afferent and then motor efferent neurons and this is kind of how they come and go through the spinal column, and you know how the brain controls it. There's three layers also I want to talk about, and these are kind of harder to see. You have the dura mater, you have the pia mater, and you have this subarachnoid space. There's three different layers of the spinal cord. So the dura, I think, is the farthest outside. It's kind of hard to tell. It's like right here. And then right inside that, you have the pia. And then you have this, actually, the pia is the most inner. And then between the pia and the dura is the arachnoid space. I would just say arachnoid space, not subarachnoid, which is kind of right here in the middle of it. Something that we probably want to go over, you know, maybe later on, but that's just some anatomy stuff. Okay, I think, yeah, and the next one is basically what I just mentioned. Okay, I think that's probably all I want to talk about tonight, and we will touch on this later on. I think we're running into 2 weeks of neurophysiology. That is enough information, I think, right now. Is there anything else that we want to talk about? Any other topics worth bringing up that we want to review or we're not quite clear on? Jessica, I did see your email. I got to figure out how we get the attendance on the spotlight sessions. I know I have to send you guys a attendance form, and then you fill it out. And I think your lecture structure, it somehow gets imported into Blackboard. I still have to clear that up. Does everybody know how you track your spotlight sessions points? Yeah, I'm not quite sure either. I may have to look it up. Yeah, I'd like to know. I'd like to know myself. I know it's what I will say is that it doesn't. It's such a very small percentage of your grade that it doesn't quite matter. It's just It's trying to encourage people to. I mean, obviously, if you're on the line, then yes, it'll move it. But you're encouraged to join these sessions and help. Yeah, man, I'm going to give you all right now I'm gonna send. Let me go find the attendance form real quick, and I think we're wrapped it up today. Okay. I don't know the answer. I just read everybody's, yeah, I'll post the video. So people ask me, how do you find my channel? I don't know. Just type in my last name, and you'll have all my stuff come up. So, you know, and then you can find the most latest one. I'll probably do it. It has to upload on to Collaborate, and then that takes a long time. And I'll probably do it around 10:30, hopefully before midnight. Yeah, you're right. You're right. Okay, Jackie. Okay. Oh yeah, thank you. You read my mind. I was about to get French, but I didn't want to embarrass myself. Yeah, Jackie. Okay. I was reading Caitlin's wording too. Yeah, I know it. That's the problem. Sometimes it kicks you out. Sometimes the internet issue. Sometimes it's the link. This is the first time that we're kind of rolling this out, so we're trying to figure it out. But anyways, without me rattling on, that is the link for the attendance form. Please fill all that out, and we will kind of go from there. And I will may email, and then next time you all join one of my sessions, I will, or Dr. McCain's session, I will email and find out how people can tell if they've satisfied their requirements or how many they need to. My guess is you probably want to be maintaining your own attendance sheet. And just to make sure, because I don't know the answer and I have to go find it, and I don't want somebody missing points. So fill that out, and then when you're finished, let me know you are finished, and I'll hang around to make sure nobody has any problems, and then we will go from there. Oh, neurophysiology with Mattmiller, or with me. Yep, Cage. We'll see you next time. Have a good week. See you, Jessica. Oh, neurophysiology, Caitlin. I'm going to spell it out. Well, I don't know, Jackie. It's a good question. I would try to attend as much of these as possible. Track on the syllabus. It sounds like you know the syllabus with the A and B portion, so you know that part. I would just try to make sure you complete. I'd make sure you try to attend 6 of them and get your name on these attendance forms. That's how we can keep track, is through the attendance forms. But I'm going to look into that a little bit harder. I may email some people and see if I have any more information to update. Then hopefully, if you want, you can email me later on and ask for an update, or I'll see you in the next Spotlight session. Yep. See you Wednesday, Amanda. I'll be there. Okay, Kayla. Yeah, we'll catch you Sunday. Sunday's always a big group. Okay. And if you keep missing me, just send me an email and see if I got any answers for you. Because I don't think you're one of mine, but maybe you are. I have so many people in the online section. All right, Jarita, thank you. All right, I think everybody's out of here.